Very good. Hello, everybody. We are continuing, of course, with the neuroanatomy course, and I hope you are enjoying the first lectures. This is the last one, this is the last lecture, which is part of the introduction to the course. It's going to be a fast moving because we have a lot of pieces to go. We're not going to spend a lot of time on details, but next week we will begin with the medulla, and then we will get into a lot of detail. So just outline is what we're doing today. We're going to look at the brainstem, the cerebellum, and the diencephalon. You may remember from our last time together that at 4 weeks of gestation, the nervous system is at the 3-vesicle stage, and a week later, it's at the 5-vesicle stage. The prosencephalon becomes the telencephalon and the diencephalon. The midbrain remains the midbrain, and the hindbrain or rhombencephalon will develop into the metencephalon and myelencephalon. We're going to be, last time we restricted ourselves to the telencephalon. Today we will take up the structures that develop from the other 4 vesicles. We'll continue to use this terminology. Mostly in the brainstem, we're going to be talking about dorsal and ventral, because it's below this flexion having to do with the cerebral hemisphere, but not entirely, and we will use the term rostral to indicate closer to the frontal pole. So the introduction here, the brainstem as a whole is bounded rostrally by the diencephalon, and the diencephalon is part of the cerebral hemispheres. The ventral surface of the brainstem is dominated by the corticofugal pathways. We're going to look at those, everything coming out of the internal capsule, basically. And then gradually, the brainstem will transition into the spinal cord. Obviously, the medulla is sitting immediately above the spinal cord, so we're going to lose the medullary pyramids. You see an anterior median fissure in the cord. Of course, the caliber and cross-section becomes reduced. The cranial nerves disappear, and in their place, they are replaced by the segmental nerves. And then the 4th ventricle will also disappear and become the central canal. We'll see this picture. Several of these pictures are going to occur several times. And each time, though, I want to keep us focused on particular elements. So here we're looking at the ventral surface of the brainstem. We can see above it some of the diencephalon. We'll come to that later. Here are the corticofugal pathways. This is the crus cerebri, the cerebral peduncles of the midbrain, that's running through and contributing to the basis pontus, the pontine nuclei, and then here are the pyramidal tracts running on the surface. These are some of the cranial nerves coming out, and here's the central root, the ventral root of cranial nerve 1. So this is going to be our transition. You can see here the decussation of the pyramids. These corticofugal pathways start with the corona radiata and then go through the internal capsule. And obviously, this massive corona radiata is not fitting into the pyramid. All right, that makes no sense. So really, that lets you know that what's happening is that at each descending axial level, there are a lot of fibers which are leaving the corona radiata, that are leaving the internal capsule, that are contributing. They're stopping off at the diencephalon. They're stopping off in the midbrain. A huge amount of them are going to cease as they get to the pons, where we're going to look at that as they create the pontine nuclei and the middle cerebellar peduncles, and then a relatively small portion continue as the lower corticobulbar tract and the corticospinal tract in the pyramids. Looking at the brainstem and its dorsal surface, we will see some of the diencephalon immediately above it. We will see the thalamus and the epithalamus, and we'll look at those. The term epithalamus we'll get into in the future, but has to do with the habenula and the pineal gland. We will look at that today, as well as the stria medullaris, which we'll also look at today. And then you get to the brainstem proper. We're going to look at the midbrain. Remember, we're looking at the dorsal surface. We're going to see the quadrigeminal plate, the superior and inferior colliculi. And then if you don't take away the cerebellum, all you're going to see is the cerebellum. But assuming that you remove the cerebellum, then you look into the floor of the fourth ventricle, where we will see the facial colliculus that Juan Solano and I were discussing at a localization session we had, as well as hypoglossal eminence and the tubercles of the cuneate and gracilis nuclei. So here we are looking at the dorsal surface, and it's dominated by the thalamus. I mean, that's all there is to it. The thalamus is massive. Here we have the epithalamus. This is the habenula. This is the pineal gland. Here we have the stria medullaris, which you can see, and you can see it in gross specimens as well. Those are diencephalic. Well, that's diencephalic. Brainstem proper, superior colliculi, superior colliculi, inferior colliculi. The fourth nerve coming out on the dorsal surface of the brainstem. And then now you can see here from the level of the pons and medulla that we've cut away the cerebellum and its associated superior and inferior medullary vellum. We'll look at all that in a minute. We're looking down into the floor of the fourth ventricle, I'm sorry about that. And you can see the facial colliculi. You can see the hypothalamus trigone. It's worth pointing out, I think, the area postrema, area which is, as you know, associated with vomiting, intense vomiting. And then we have the tubercles of nucleus cuneatus and gracilis, which end the posterior columns and give rise to the secondary neuron heading up to the thalamus as part of the medial meniscus. If we look at if we switch from these gross external views and start looking in cut sections of axial, then we start to get a sense of the ventral to dorsal transitions within the brainstem. In general, the most ventral portion of each portion, midbrain pons and medulla, are all the ventral fiber bundles that we talked about. And they have different names depending on where they are, but they're all that one pathway. Immediately dorsal to that will be the tegmentum, which is the central core of each of the sections. The reticular formation runs throughout the tegmentum. But so do specific cranial nerve nuclei, which obviously depend on whether you're in the midbrain, the pons, or the medulla. And then as the most dorsal portion of each of the brainstem, you have what's called the tectum, or el techo in Espanol, the tectum, which is the roof. All right. It's really only seen in the midbrain, where it is the quadrigeminal plate. In the hindbrain, the rhombencephalon, whether midbrain or, whether pons or medulla, it is represented by the telochoroidea and the cerebellum. And we're going to look at all of this. So here you have a midline sagittal view. We're going to look at this view several times. Here you have, again, midbrain, pons, medulla. On the surface, you know, is the crus cerebri, the basis pontus, and the pyramids. This bulb is from the olivary nuclei as well, we'll look at. On the dorsal surface, sorry, I'm going to ignore that. On the dorsal surface, excuse me. On the dorsal surface is here the tectum. So the rest of this, of course, is tegmentum running through the central core. And then you have the tectum. And the tectum is a quadrigeminal plate here, dorsal to the aqueduct of Sylvius. And then you have here the fourth ventricle, and this is going to be the superior medullary vellum, the inferior medullary vellum, which will also give rise to the choroid plexus, and we'll look at that in a minute. And then the huge cerebellum, which we will look at in detail. Again, we're going to see this view multiple times. This is the Weigert stain from the Yakovlev collection in a sagittal view of the brainstem. And clearly the basis pontus dominates your attention in this view, as well as the cerebellum. But you can see here the midbrain. This is the midbrain transition into the hypothalamus. This is the mammillary body. This is the hummingbird view that we know and love. The red nucleus is sitting in here, the decussations of the superior cerebellar peduncles. We're going to look at all of that. and then cranial nerves and reticular formation traveling through. Here's a bit of the olive in the medulla. And you can see the superior medullary venom. The only thing other than containing the fourth ventricle, the only other thing that's interesting there is that the fourth cranial nerve is going to travel through that. When we this is by no means at the detail that we're going to consider next week. But if we now start to look at the various portions of the brainstem, we know that the medulla has its characteristic appearance. This is the most typical view that we see. You have the pyramids up here, you have the fourth ventricle back here, all right? And these eminences, which are due to the inferior olive. This is what makes the medulla look like the medulla. But there are also these very important inferior cerebellar peduncles, which are going to be coming up. And that, when we look at the medulla next week, we will divide into both the restiform and the juxtarestiform bodies, looking at different information, eventually entering into the vestibular system and the cerebellum. And then, of course, in the medulla, we have cranial nerves 9 through 12. And 8 sits sort of at the junction, and we'll talk about that later. Focusing on the medulla, right? So here we have various cranial nerves emerging, everything from glossopharyngeal to hypoglossal, vagus as well. We see the pyramidal tract. We see the tubercle of the inferior olive, right? And so this is all medulla down here, and another view of cuneate and gracilis nuclei on the dorsal surface. I put this red line in here as a rough landmark between pyramid and spinal cord. And again, what we're looking at here is just the medulla, and we see the pyramid, the tubercles, and the various cranial nerve emerging. And the colored view, same thing, giving you a sense of how crowded this emergence is. We'll get to these. This is 7 and 8, cranial nerve 6, 7, and 8, which emerge at the pontomedullary junction. We'll look at that in the detail. This left picture is an important picture that we're going to deal with next week, but it introduces a very important concept. And that is that there is a columnar organization of the midbrain, of the brainstem in general, where close to the midline you have the somatic motor nuclei, cranial nerves 3, 6, 12, 4. Those are true somatic motor. And those are the only ones that are true somatic motor. And then paramedian, right off the midline, are going to be the motor nuclei of the branchial arch. Those are going to be the trigeminal and facial nuclei, which are derived from first and second branchial arch. And then you're going to get into visceral motor, which will be vagus and things like that. And then, and I don't like the order that they put these, but then you're going to go transition into visceral sensory, general sensory, and special sensory. And by special sensory, we have hearing and vestibular. So this is a very important organizational principle. The second thing that I want you to take away from these pair of diagrams is how long some of these cranial nerve nuclei are. And let's focus, we're in the medulla, let's focus on the hypoglossal nucleus here for a moment. Why you're going to see the hypoglossal nucleus when we look at a bunch of axial views everywhere from the caudal medulla all the way up to the pons. All right, that's real. It's not like these, like say the abducens, which is a more or less a globular nucleus, or the facial nucleus, which is a discrete globular nucleus. This is elongated, and so are so is the vagus complex. And look here at the solitary tract is huge. All right, and we're going to be dealing with that in the weeks to come. Again, from the Yakovlev collection, a view of the medulla. I told you this section can be expanded almost ad lib. We're in the medulla, a very caudal medulla. Starting at the ventral surface, you have the arcuate nucleus, which is going to blow up in the pons into the pontine nuclei. We have the pyramidal tracts, which are well seen and very discrete here. The purse string of the inferior olivary nucleus. And then here you have the decussation of the medial lemniscus. Here you have the MLF. And then back here you have, well, nucleus gracilis, nucleus cuneatus, a whole lot of trigeminal complex and vagus complex, and the nuclei that are going to hover around the central canal and eventually the fourth ventricle. Again, we'll get to the detail next week. Nissl stain showing you the neurons that live within these bodies that we just talked about. And I've put this labeled low mag up here for you to use. Remember, let me take this opportunity to remind you that I am expecting you to spend an hour in addition to going through the talk with me, an additional hour every week in order to make yourself comfortable with all of this huge amount of information. As we move to the superior or most rostral portion of the medulla, we start to see hanging out sort of the panniculus, the belly fat of the basis pontus hanging over. And so the arcuate is going to, like I said, just explode into these pontine nuclei as we move rostrally, dwarfing the inferior olive and everything else that we've seen in here. And of course, as we move rostral, the fourth ventricle is going to expand. What does this say? This is the fourth ventricle. I can't see the top of it, but from the fourth ventricle. So we're going to spend a moment here and reorient ourselves to the gross structure of the fourth ventricle. Fourth ventricle runs from the central canal to the aqueduct. The transition from fourth ventricle to aqueduct is often referred to as the fastigium. The roof of the cerebellum and superior and inferior medullary villi, so the roof of the ventricle is made by those, as we said. And the inferior medullary vellum is of interest because both that it gives rise to the choroid plexus, and you remember that from our earlier lecture a couple of weeks ago. And then also that the foramina of Majandi and Luschka emerge from the inferior medullary vellum. The widest portion of the fourth ventricle is immediately caudal to the middle of the cerebellum peduncles and has a lateral recess. We'll look at that. And we've already looked at this, but if you pull off the cerebellum and the belly, then you end up looking down into the floor. And like I said, we've already dealt with that. Just to focus for a minute, here we're looking at the dorsal surface, and here's the superior medullary vellum, the inferior medullary vellum, and obviously the cerebellum. And here's the fourth ventricle in the common shape that we know from sagittal MRI or CT plane. And here we are at that level of maximum expansion where you also have these lateral recesses. And we're just looking at the ventricle immediately bounded posteriorly by vermis and anteriorly by the floor of the fourth in the pons and medulla. And we've already dealt with this picture, but just to review it. Okay, moving into the pons. The basis pontus dominates everything about the brainstem and obviously dominates the pons. The basis contains the descending corticospinal and corticobulbar pathways, as well as the corticopontine pathways, which are the largest by far. And those corticopontine emerging from cortex are going to synapse on nuclei, on neurons within the cerebellar nuclei. And it's huge, and that's what gives the basis pontus its striated appearance on the axial views, as well as the transverse fiber bundles, which are going to coalesce to become the middle cerebellar peduncles. In the tegmentum of the pons, you have, as always, reticular formation, but you also have cranial nerves 6 through 8 are in the caudal portion of the pons, and up in the mid portion of the pons will be cranial nerve 5, which is going to exit right through the middle cerebellar peduncle. So here you have this view that we keep using, and you can now see down, here's cranial nerve 6, here's cranial nerve 7 and 8, here's cranial nerve 5, this massive trigeminal structure, which is just penetrating right through the transverse fibers of the pontine nuclear basis pontus. again, just making you aware of how big the trigeminal complex is, color-coded to help you identify the nerves emitting. And we've already done this. I want, however, to point out, in addition to cranial nerve 6, cranial nerve 7 nuclei, cranial nerve 5 motor nucleus, and the huge and extensive sensory path nuclei that is the principal nucleus of the trigeminal nerve. And it's just huge. It runs well into the midbrain. It goes all the way down into the spinal cord. Most of it is collected, though, as a principal sensory nucleus in the palms. And you can see some of these pathways. This is interesting. Again, the facial colliculus occurs because of the movement of the emerging cranial nerve 7 nerve, which goes right around the cranial nerve 6 nucleus. Back to this picture, which we've seen before, just to show you, again, we are now in the pons. We're looking at basis pontus in a close to the midline view, transverse fibers going across corticobulbar tract, corticospinal tract running through, cranial nerve nuclei aligning themselves in the tegmentum, all the way through here, and the reticular formation. And then you can see the same thing here on the Nissl stain. When we look in the axial view, we can see the fourth ventricle, obviously, but we want to focus on basis pontus, transverse fibers, an enormous amount of nuclei. I look down at the Nissl to see how many neurons and neuronal collections are out here, and then the tegmentum of the pons with its reticular formation and the cranial nerve nuclei restricted to this small area on top of this massive area that will give rise to the middle cerebellar peduncles. Again, I cannot see what the title of this slide is because of Zoom. We are right. So this is cerebellum, and we know that the cerebellum is derived from the metencephalon, not the myelencephalon, but from the metencephalon, and not the mesencephalon either, for that matter. The shape of the cerebellum grossly is just basically because it fills the entire posterior fossa. It just fills it. And it takes on the shape of the posterior fossa, which is a combination of bone and tentorium. And it just fills it, and that's all there is to it. Grossly, you can divide the cerebellum into the vermis and the cerebellar hemispheres, but I got to tell you that the hemispheric landmarks of the cerebellum are notoriously useless. They are not very helpful at all. That's not how the cerebellum is organized, and it's just what we see it from the outside. The cerebellum, and obviously we're going to spend a lot of time on cerebellum later in the course, but it is organized suprasegmentally. It really doesn't care that much about quite the sensory information or what part of the brain it's projecting to. I mean, it does, but not the cortical structure in the hemispheres. These are all going to emerge when we get to the level of the cerebellar nuclei, which we're going to look at shortly. You should not consider the cerebellum part of the brainstem. I don't think that works. It is really, it's related to every portion of the brain. And I just think that you should think of it as a suprasegmental. It's above all of that, calculating, coordinating. It's a marvelous structure that we will consider in detail later. Here it is sitting in place, taking up its role behind above the occipital bone and below the tentorium. Until you get to embryology, the organization of the cerebellum is very difficult, I think, to understand, because you start seeing things like the nodulus and the floccular portion sitting somehow above the lateral portions, which it makes no sense. And it doesn't make sense. In fact, it won't make sense until we look at the embryology. Then it will make perfect sense, because you have this huge flexion which is going to occur in the creation of the cerebellum. And with that flexion, you realize why the most rostral and the most caudal section are next to one another. And you're going to see that multiple times in the development of the nervous system, especially with the limbic system and that sort of thing. But until you get to embryology, let's just put it this way, when we get to the embryology, it will make much more sense. Meanwhile, here's the anterior cerebellum, here's the Posterior cerebellum, this is the primary sulcus, and we'll look at sulci, but they're not really useful. All of that folia that you see, those striations, those folia they're transverse. There are numerous folia. One of the things that's really interesting about the cerebellum and contributing to this notion of suprasegmental structure is there's no midline raphe. It's the only part of the nervous system which really doesn't reflect across the midline, and it doesn't. It's just continuous across. You have these 5 horizontal fissures, which, you know, you can poke your finger in if you want. They don't contribute much to the organization. The primary one defines everything rostral to the primary fissures, the anterior lobe, everything caudal, if you will, to the posterolateral fissure is or everything between the primary and posterolateral fissure is the posterior lobe, which will include the tonsil. And so when you look at it this way, this just doesn't do me very much. I don't think it's worth spending a lot of time on it. There's your primary fissure. The posterolateral fissure is down here. There's a nodulus. Obviously, we're at the level of the vermis here. All of this, including the tonsils, is all the posterior lobe of the brain, anterior, posterior, floccular, nodular, that sort of thing. It's just not the right way to think about it. The cerebellum is organized more along the lines of midline, being where the vermis is going to be for gait and station. Paramedian is going to be having to do with legs, the lower half of the body, things like gait, ambulation, kicking a soccer ball, stuff like that. And then the paired flocculi, the paired lateral hemispheres are for hand, for cognitive, things like that. Don't think of it as only motor. Cerebellum, not only motor. There's a lot of huge amount of cognitive function in there, as I'll show you in a minute. A better way to think about the cerebellum and the growth structures is the floccular nodular lobe is the older portion of the cerebellum, the archicerebellum. The nodulus in particular is going to be related to the vestibular pathways, okay, and contribute neck control to eye movements, stuff like that. We got to integrate all that stuff. The paired flocculi on either side, similarly, are going to be almost vestibular in nature, more than cerebellar, if you will, in nature, and they're going to play a major role in eye movement, and we'll deal with it later. So nodulus, floccular. Here are the tonsils, which are part of the posterior lobe, anterior lobe up here. Again, that, you know. Here, I put this picture in here. I just, yesterday, I found this nice. I was wondering myself, here's some from MRI data, and you can see some of the areas of the lateral hemispheres which are involved in spatial orientation and language, okay, and things like working memory, okay, as well as sensory motor pathways in the paramedian portion of the cerebellum. And so I put this up there, and you can look at this at your leisure. But there is a topography to the cerebellum. I'm not saying it's all one gross massive computer, which is sort of what it is, but different parts of the computer seem to be focused on different functions. It's important when we talk about the cerebellum to deal with the cerebellar nuclei. These are often referred to as the deep nuclei. A simple mnemonic aid is that they fit in the DEFG. They're all dentane, emboliform, fastigial, and globose. However, I don't like the organization of the DEFG because they don't quite follow that way. The dentate is by far the easiest to see. It's the largest. You can't miss it when you look at a cut section of the middle cerebellar peduncle because it's sitting right in the middle of it, okay? The vestigial nucleus is in the floor of the fourth and is the one that's most closely related to projections from the vermis, and therefore is going to be gait and station and things like that. And as you can imagine, will relate to the floccular nodular lobe. And then the emboliform, together with the globose nucleus, make up the nucleus interpositus. You really only separate emboliform and globose in primates. In almost all other animals, they're together. They're very difficult to separate. And when they are together, they are known as the nucleus interpositus. Again, here's a sagittal view. We are now very much off the midline, and you can see the pontine nuclei. You get a much better sense of their longitudinal rostrocaudal extent, which you have to integrate with that axial view that we saw. You just have to get that straight. We're already now looking at the middle cerebellar peduncle, and as I said, sitting within the middle cerebellar peduncle, the dentate nuclei, which are going to give rise to the superior cerebellar peduncle. Nice view of the Nissl stain. And when you look at this and you look at the cerebellum, we'll look at the cerebellum, like I said, in the future. But I want you to realize that this deep violet staining on the Nissl is because half of all the neurons in the brain are in the cerebellum. So you need to put that in perspective. And when you look here, so when you see this foliar structure, whether you're looking at the Weigerts and the myelinated, which creates these beautiful patterns, almost what do you call them, Mandelbrot type of patterns. You see here this just exquisite organization of the cerebellum that we will delight in studying in a separate lecture. Same thing here. Look at it. So we're looking at the dentate here, but just look at the Nissl stain of the cerebellum. It's impressive once you realize what it is. Here's the dentate nucleus, the most lateral. Here's the fastigial nucleus, the most medial. And here are the emboliform and globose together, creating the nucleus interpositus. So now what? We're in the midbrain. In the midbrain, we will look at the tectum, which is really only developed in the midbrain. We're going to, the superior colliculi and the pretectal area are all visual. The inferior colliculus is all auditory. When we study the systems, when we study the midbrain, when we study the auditory and visual systems in neuroanatomy, you're going to see that although the superior colliculus and inferior colliculus sit next to one another, they are not analogous to one another. They are very, very, very different in their functions within these two major special sensory pathways. And we're going to deal with that. But I want to dissolve that idea that they're going to be analogous to one another because they have similar names, sit next to one another. They don't. All right, the auditory pathway is extremely different than the visual pathway, and we will highlight those differences later, of course. Within the midbrain, of course, is cranial nerves 3 and 4, which we'll look at. We know 4 exits dorsally. We're going to look at the crus cerebrae, which we've discussed, as well as the superior cerebellar peduncle, the substantia nigra, and the reticular formation. In this view, we're back to the midbrain, here dominated by the huge crus cerebrae, knowing that that will not fit in the pyramid, and so it must be giving off massive fibers within the spaces pontus. Here you see emerging the cranial nerve 3, mammillary body sitting here at diencephalic. Okay, here we have the colliculi, and notice how closely related we are to the medial geniculate and the lateral geniculate, diencephalic structures for sure, that are closely related to superior and inferior colliculi. All right, and the emerging 4th cranial nerve coming off the back. And in color, the same thing. And then these views, and now you can see up here the column of the oculomotor nucleus, Edinger-Westphal nucleus, trochlear nucleus sitting below it, separated almost by accident, if you will, emerging on the posterior, emerging in the midline, because these are true somatic motor nuclei. On the dorsal surface, and we're only looking at the midbrain, so the superior colliculi, inferior colliculi making a quadrigeminal plate, and the 4th nerve emerging in the superior medullary vein. Almost starting to make sense, right? Looking here at the this off midline view, we can see the crus cerebrae, we can see the longitudinal extent of the substantia nigra. I think it's so important to understand that view that we get, it looks like of the substantia nigra, which makes it look like a flipper in a you know, what do you call them, in the arcade game, pinball machine. It's not a flipper. Okay, it's a long extent, and we see it in cross section all the time, but it runs the whole length of the midbrain, and really it overlaps into diencephalic structures. And so now we're on axial view here, and you can see here's the midbrain, obviously the crus cerebri, the substantia nigra, the red nucleus and its surrounded pathways, some of the tectum, right, the tectum of the midbrain, periaqueductal gray, the tegmentum of the midbrain, and then the tectum of the midbrain back here, which will be the colliculi. The fastigium, as the fourth ventricle, is going to become the aqueduct of Sylvius. The interpeduncular fossa, okay, separated or divided by this pathway, which is going to the mammillothalamic tract here. All right, we're going to study this in the future, but that's diencephalic. Up a little bit higher, as we are, excuse me, a little bit lower, we have the crus cerebri, right? The substantia nigra. These are mammillary bodies, the interpeduncular fossa. Here we are at the decussation of the superior cerebellar peduncle. Other pathways related to the medial lemniscus coming up. The MLF is still present, periaqueductal gray, all of that there. Emerging third nerve nuclei, third nerve structure itself. And just to go back here, just to sort of showing you the Nisselstein view of the pars compacta. Those are the big neurons that you can see from the substantia nigra that runs as a sheet. All right, transitioning finally up into the diencephalon. So the diencephalon as a whole, we're going to break that up into pieces. The diencephalon is housed within the cerebral hemispheres. The diencephalon consists of paired structures on either side of the third ventricle going out to the internal capsule. So the entire diencephalon will sit between the third ventricle and the internal capsule. Those are the medial and lateral borders. When you look at the rostral border, it's going to be, or first of all, the most rostral border, yes, will be bounded by the lateral ventricles, the corpus callosum, the fornix, and the velum interpositum. The inferior border is going to run right into the midbrain. It's going to run right into the tegmentum of the midbrain. The most rostral border will be the foramen of Monro. And we will look at the four divisions of the diencephalon: the epithalamus, the thalamus, the hypothalamus, and the subthalamus. All right, a large coronal view of the cerebral hemisphere with a little bit of midbrain attached. And you can see the third ventricle, obviously. This will be hypothalamus here. Here's the thalami. All right, here's the subthalamic nucleus. This is the area known as the zona incerta. We'll look at this in a minute. Here's the pars reticulata of the substantia nigra and the crus cerebri. All right, and there you are. And then you can see the cerebellum, right? You can see the lateral hemisphere, the lateral ventricles. This is a bit of caudate, right? Here's the internal capsule that makes the lateral border. All right. Focusing for a moment on the epithalamus, which we defined as having the habenula and the pineal gland. The habenula is a structure studied by one of my mentors, Nick Len, taught me a lot of pediatric neurology as well as a lot of anatomy. Next studied Dr. Len studied the habenula. It appears to integrate limbic influences and contribute those to the midbrain and substantia nigra. It receives input from a lot of structures in the diencephalon, the septum, the preoptic pathways, the anterior thalamus, and the globus pallidus. The septum is interesting. I spent some time this week trying to research the septum and figuring out what its embryologic origin was for the purpose of this lecture. And the answer is, at the end of all this search, I have no idea, because neither does anybody else. They simply don't know where the neurons come from. Given its position, it seems to be diencephalic, but I don't know that. Like I said, it is simply not known. The pineal gland is the posterior or dorsal extension of the habenula, and of course is in both birds and in humans, has to do with circadian rhythms. In humans, it's almost a vestigial structure. The stria medullaris takes information from the septum and transfers it over to the habenula. So this is all an integrated structure. So here's your epithalamus, the habenula, the pineal gland, and the stria medullaris. This is the epithalamus, the highest level of the diencephalon. The thalamus, as you all know, I believe is the key to understanding the central nervous system, and we will spend a lot of time looking at the thalamus. The thalamus distributes nearly all afferents, except for olfactory, you can say all afferents, to the cerebral cortex. It integrates motor information. It contributes to the Brodmann's area 4, and thereby running the corticospinal tract. It activates and regulates virtually all of the electrocortical activity throughout the cerebral hemispheres. There is a massa intermedia, that is, the thalamus crosses the midline in about 80% of humans. Sometimes it's small, sometimes it's huge. Sometimes you can barely see a separation between the right and left side. It doesn't seem, whether it does that or not, doesn't seem to correlate with very much. The thalamus can itself then be, well, it can be infinitely divided, but at the gross level, you can look at the central nuclear groups, and you can look at the extensions out of the central thalamic nuclei. And a lot of people don't want to consider the anterior tubercle or the metathalamus as part of the thalamus proper. The pulvinar, the medial and lateral geniculates are in many ways separate, but in many ways share so many of the functional organization and local neuronal structures of the central nuclei that others would say it makes no sense to separate them. But there you are. They can be separated if you want to. And then within the thalami are the internal medullary laminae and all of their associated nuclei, which we will deal with in detail. This picture I'm just showing you to give you a sense of the relationship of the thalamus to the other portions of the cerebral hemisphere. And there is a very strong topology relating the thalamus and the thalamic nuclei to the portions of the cerebral hemispheres. Looking again, just back to the gross view, and again, I think the first thing that catches your attention is how big the crus cerebri are, the cerebral peduncles, but also how big the central nuclei of the thalamus is. Here's the pulvinar sitting out behind. Here's the optic nerve, which is strongly diencephalic. Here's the lateral geniculate receiving the optic tract, right? Here's all of the caliculi that we've already previously mentioned, pulvinar sitting posteriorly from the Yakovlev Halim collection. Let's again focus on boundaries for the moment. Third ventricle, internal capsule. Right? Everything between the third ventricle and the internal capsule is diencephalic, or the entire diencephalon sits between them. Here's the anterior. You thought, I don't know, how is this related to the foramen of Monro? That is a little bit tricky, I think, in 3 dimensions. But here in the slice, you can see it, because here's foramen of Monro. Let me go back to that, sorry. So third ventricle and foramen of Monro. So, okay, and then here's septum, here's fornix, but that's going to develop into septum. Thalamus, pulvinar, central nuclei, okay, yeah, I could see this, okay. A little bit of pineal gland here. Again, a little bit lower, and here you have the anterior commissure connecting the 2 temporal lobes. You have third ventricle, right, and again, thalamic nuclei, internal capsule. All right, so down here showing you the caliculi. Back to that coronal view that we had before, but now understanding a little bit more and seeing again the central nuclei of the thalamus and looking down into lower portions, which we will in a minute. Still focusing on the thalamus itself and how huge the pulvinar really is. Pulvinar is going to have to do with integration of visual information and contributes to language. Middle cerebellar peduncle, just going, here I am, how huge it is and how just gorgeous the structure is. Here's the dentate sitting in the middle of it. So again, we're just looking at gross stuff today. We're going to return to all of this. Don't let all this stuff overwhelm you. It shouldn't at this level. We're just looking at the overall organization. So when we get into the weeds, when we start bumping into trees, we're going to have a sense of the forest. Today, there's a sense of the forest of the diencephalon, brainstem, and cerebellum. So finally, getting to the end of this, the hypothalamus. It is ventral to the hypothalamic ventral, to the hypothalamic sulcus. We'll look at that. It forms the inferior and lateral walls of the third ventricle. Get used to that notion. Associate the hypothalamus with the third ventricle because the walls of the third are the hypothalamus. It's going to run from the optic chiasm all the way to the mammillary bodies. We're going to look at some growth structures in a minute of the inferior or ventral portion, if you will, of the hypothalamus, the tuberosinium, infundibulum, mammillary bodies, that sort of thing. Within it will be the fornix, and then of course you know the functions, but we will look at the functions later because we look at the divisions of the hypothalamus into all these different endocrinologic visceral functions that are so important. Here's the optic chiasm, here's the mammillary bodies, here's the tuberosinium, which is then going to give rise to the pituitary stalk, all right, optic tract, right? And we'll come to pons. We'll come to all these structures, olfactory, trigone, that sort of thing. I love this view. I want to spend a minute on this view because this is a very difficult area, at least for me, to integrate in 3 dimensions. And this picture really helps. So here's basis pontus, right? Here's the tegmentum of the midbrain, right? Here's a mammillary body. Here's I don't care if you want to call this the lateral ventricle or the wall of the hypothalamus, I don't care. Here's the mammillary body. Here's the lamina terminalis, okay? Here is I can't see, here's foramen of Monroe, and I have to figure out what this is. This would be optic, so here's the optic tract, this is mammillary body, I guess. No, I don't like that, this is mammillary body. Thank you. What is 6? I can't see what is 6. This is I know. This is the mammillothalamic tract here. Here 5. This is the massa intermedia of the thalamus. Here's the posterior portion of the hypothalamus, or the third ventricle, whichever you want. And here's the pineal gland. So a very useful, this structure is vascular. Wow, we're here to look at the vascular supply of the diencephalon and of the midbrain, all these tangle of vessels coming off the basilar artery. Another coronal view down emphasizing how the hypothalamus is the wall of the third ventricle. Here's the optic chiasm. All right, infundibulum coming off right behind it, heading down into the pituitary. And some of these other pathways, which we've considered in the past and will consider at a later time. We end with the subthalamus, which is obviously under the thalamus, the hypothalamus below the thalamus, the subthalamus under the thalamus. It is both ventral and ventral to the thalamus and lateral to the hypothalamus. And for the most part, it contains the subthalamic nucleus, but it's also traversed by a lot of fibers which give rise to this white matter region, collectively known as the zona incerta. So here's the zona incerta. Here is the subthalamic nucleus and its intimate relationship to pars compacta of the substantia nigra is obvious here as it virtually descends into midbrain, or this ascends up into the diencephalon. That's a mighty tight relationship there. And that's it. So a quick tour. Let me end sharing here. Stop sharing. All right, so a quick tour of the brainstem, cerebellum, and diencephalon. An incredible amount of neuroanatomy in there. We're going to be returning to see those starting next week, and when we will consider them in detail. So before we end, are there any questions from anyone sitting in?